Hello everyone, myself Sritama Deb from Shoolini University, and my topic for the project was molecular docking study of routine ligand with the target protein interleukin 6 IP9M. So first, the background. Drug discovery is a process which involves identifying the molecule that can bind to specific disease-related protein. Traditional experimental screening of thousands of molecules require large time, cost, and laboratory effort. Computational approach such as the molecular docking allows researchers to predict the ligand-protein interaction before performing any experiment. This reduces the experimental workload and helps shortlist promising compound for further test. So, what is molecular docking? Molecular docking is a structure-based computational method used to predict how ligand fits into the binding site of a protein. The process evaluates both the geometry of binding and the interaction energy. Docking software generally generates multiple possible ligand orientation known as the poses. Each pose is scored based on the predicted binding energy. A ligand can be docked in the active site of the protein in different orientations, which are known as the poses. More negative binding energy value indicates strong and more stable interaction between the ligand and the protein. Next, my target protein, which was IP9M interleukin 6. The three-dimensional structure of protein IP9M was obtained from the protein data. This protein has defined active site suitable for ligand binding. Studying ligand interaction with this protein can provide insights into possible inhibited molecules. Understanding how small molecules interact with the protein help in identifying candidate for drug development. So this is my target protein, which is responsible for the disease Parkinson. So let's now talk about the ligand. Ligand information routine. Rutin, the PubChem ID has been provided, is a natural flavonoid compound widely found in plants. It possesses several biological activities such as antioxidant and anti-inflammatory properties. Structurally, rutin contains multiple hydroxyl groups capable of forming hydrogen bonds. These features make rutin suitable for docking studies because such functional groups can strongly interact with the protein residues. So this is the ligand. Aim and the objective. The aim of this study is to analyze the binding interaction between rutin and the protein. Objective include preparing both protein and ligand structures, performing docking simulation, calculating the binding affinity values, and analyzing interaction types inside the protein binding pocket. The study also aims to evaluate whether rutin shows characteristics of potential inhibitor. Software and tools used. PyRx was used as a main docking platform because it integrates docking and visualization tool in one interface, and also PyRx is the most simplest tool used for docking. AutoDock Vina was used as the docking engine because it predicts ligand orientation, calculates the binding affinity using a function scoring. Discovery Studio Visualizer was used to obtain the dot complex and analyze hydrogen bond and other interactions. Protein and ligand structures were obtained from Protein Data Bank and PubChem respectively. Protein was taken in .pdb format and the ligand was taken in SDF format. Protein preparation. The protein structure downloaded from the database initially contained water molecule and some unwanted ligands. These components were removed to prevent interference during the docking. Polar hydrogen was added to allow proper hydrogen bond formation during interaction analysis. The prepared protein structure was then converted into docking format to make it compatible with the docking software. The docking format was a PDBQT format, and the whole process of removing the water molecule unwanted ligands and then adding the hydrogen was done in Discovery Studio. Next is the ligand preparation. The ligand routine was downloaded in the structure standard structure format and converted into a three-dimensional structure. Energy minimization was performed to obtain a stable conformation with minimal internal strain. Rotatable bonds were defined so that the ligand could explore flexible orientation during docking. The optimized ligand structure was saved in docking format for use in the docking simulation, which was done in the PyRx itself. Now the docking procedure. The prepared protein and ligand was loaded in the docking software, which is PyRx. A grid box was placed around the active site of the protein to define the search region for ligand binding. Docking simulation was then run using AutoDock Vina. The software was generated as several binding poses with different energies and orientation. The pose with the lowest binding energy was selected as the best prescribed binding conformation. I have here attached the protein ligand docked in the PyRx. Here we can see the ligand has been, the best pose ligand has been docked with the required protein. Now the docking results. The docking simulation procedure produced multiple poses of protein with the protein binding pocket. The best binding affinity obtained was minus 9.3 kilocalorie per mole, which correspond to mode 0. Other poses also showed favorable energies between minus 8.6 to minus 8.1, which is quite good. The RMSD value, the root mean square deviation value of the best pose was 0, indicating it is the most stable predicted conformation. The values indicate strong binding compatibility between routine and the protein. Visualization of the dot complex showed that routine is located. Which, Visualization of the dot complex showed that routine is located inside the protein binding cavity. The ligand fits well with the active pocket without any static clashes. Its orientation allows interaction with several surrounding amino acid residues. This confirmed that the dot result is structurally reasonable. So there is the bond length of the interaction showed in the BioVia Discovery Studio, and these are the type of the interaction: conventional, hydrogen bond, pi sigma, alkali, pi donor, carbon atom, conventional, etc. This is a 2D map of interaction which shows conventional hydrogen bond, carbon hydrogen bond, pi donor hydrogen bond, pi sigma, and alkali. And this is the 3D map of interaction where blue color shows the ligand, and these are attached to different amino acids which are shown here: phenyl, leucine, arginine, glycine. And this is a 3D visualization of protein ligand complex in the BioBio Discovery Studio. So let's now do the interaction analysis. The interaction diagram shows that routine forms hydrogen bonds with the nearby residues in the binding pocket. Hydrogen bond help anchor the ligand firmly and contribute to the binding specificity. Hydrophobic interactions are also observed between the aromatic ring of the routine and non-polar residues of the protein. These interactions reduce ligand movement and increase the stability of the complex. The combination of hydrogen bonding and hydrophobic contacts indicates strong ligand-protein interaction. And the interpretation of the result. The binding energy of minus 9.3 kilocalorie mole suggests a strong and stable interaction between routine and the protein. In docking studies, values below minus 7 kilocalorie per mole indicates good binding, while value below minus 9 indicate very strong binding energy. The presence of several poses with similar energy suggests that ligand is structurally compatible with the binding pocket. The observed interaction confirmed that routine is not randomly attached but specifically oriented within the active site. This suggests that routine may interfere with protein function and act as a potential inhibitor. Now let's come to the conclusion part. The docking study successfully evaluated the interaction between routine and the protein 1p9, which is interleukin 6. Routine showed strong binding affinity and stable interaction pattern. These results indicate that routine has potential as a bioactive inhibitor candidate. Further laboratory studies are needed to validate the computational findings. Future scope. Molecular dynamic simulation can be performed to check stability over time. Binding free energy calculation can provide more accurate interaction strength. Biological experiments can confirm inhibitor activity. Structural modification studies may improve the ligand potency. I want to add about IL-6 is that it is a key mediator of increased immune response, inflammation, and hematopoiesis, and routine bind to it, which acts as an inhibitor to reduce inflammation and the heightened immune response. Thank you so much.